right. Well, good afternoon, everybody, and welcome to Bayer AgConnect for June of 2021. For those of you who don't know me, my name is Phil Taylor with Bayer Open Innovation and Strategic Partnerships based in St. Louis. And it's my pleasure to play MC and host this afternoon for what I hope you'll agree with me is an exciting conversation that we'll have here over the next hour and a half or so. We're really excited for today's event that we've been able to partner with our good friends at the University of Arizona. And we're going to get a chance to hear from them in a little bit, but also hear some of the cool things that are going on within Bayer and some of our other ecosystem partners. So if we're all ready, we'll just jump right in. But I would certainly encourage you as we go through today, if you have questions or comments or feedback, please do drop them in the chat, either for everybody or send them to me. And would love to make this event as useful for as many of you as we can. So today, we're really focused on a lot of great things going on in Arizona. As I said, Alex Strong and his team from the University of Arizona with us today, and they're going to talk us through some of the great work going on there and some of the great science and great opportunities that they have going on. Then we'll turn a little bit more inward. Bayer opened a state-of-the-art facility in Marana, Arizona, a little bit over a year ago. Basically, the last thing most of us did before COVID hit was open up our Marana Greenhouse. And Stephanie and her team down there in Marana are going to take us around and show us some of the great science and innovation that's going on at the site there, as they really think about the future of corn and corn production for our business and for agriculture. And then last, I'm really excited that Dan and his team from Space for Food were able to join us today. This is a great initiative that started up recently. Really does tie into the indoor agriculture controlled environment theme that you're going to see running through the session today. So we're going to hear a little bit from Dan and his team towards the end of the session on what they're up to. So I'm really excited about that. So those are our three main topics. I hope you'll stick around and enjoy the conversation. And as I say, please put your thoughts, feedback, comments in the chat. Would love to hear from you. This is designed to be very informal. If we were doing this in person, we'd all be wandering around some space somewhere with a beer in our hands, enjoying each other's company. And I know we all look forward to doing that again, but we will do that soon. Before I turn it over to Alex and his team, I did promise in the sort of details for this event, here's a little update from the Bayer side. In the next couple of weeks, we'll be launching this year's Grants for Ag program. And for those of you who haven't seen this before, this is our open innovation program that we run every year that's designed really to support early innovations and early innovators in taking that first step into applying a technology and thinking about its application into agriculture. And so as an organization, we provide small grants. These are typically $10,000 grants to applicants to allow them to do that first proof of concept, that first translational experiment that helps them get their idea either from the back of a napkin into the lab or even from the lab out into the field, and really do the kinds of experiments that could help them think about whether it's a project or an invention or innovation that could really impact agriculture. So keep your eye out for this. We'll talk about it much more at our event next month. But I'm as someone who's been part of this program for a couple of years now, pretty excited to see this coming in the next couple of weeks, is our hope, right after 4th of July weekend. And so with that, without sort of further ado, it's my pleasure to turn the floor over to Alex and his team at the University of Arizona. Alex.
Thanks, Phil. It's really charitable to describe the folks from U of A here as on my team, but I'm a part of the one small part of the larger College of Agriculture and Life Sciences machine here at the U of A, and I think I speak for all of us when I say that we're just really excited to be part of today's event. Thank you so much for inviting us. Like you said, my name is Alex. I work in Cal's Advancement, Advocacy, Alumni Affairs, and Corporate Relations. And without much further ado, I'll turn it over to two of our all-star faculty. On the line today, we've got Dr. Parker Anton, who's our Associate Vice President for Research in the Division of Agriculture, Life and Veterinary Sciences, and Cooperative Extension. And later, we'll hear from Dr. Murat Kasira, who heads up our Controlled Environment Agriculture Center. So Parker, take it away if you would.
Thanks, Alex, and thanks, Phil and Dan. It's great to be here to present some of the ongoing research in the college and the division. And I'm going to necessarily present sort of a certain key, what I think are perhaps key initiatives that we have ongoing without having known ahead of time exactly where this was heading. So without any delay, because a lot to say, I'll just get started. I'll start sharing screen. Hang on just a second. Okay, should be working. Okay, so as Alex said, I am Associate Vice President for Research. That says Vice President. That may be aspirational. I'm not sure there. That's a typo. And Associate Dean for Research. And what I'd like to do to start with, because the University of Arizona is certainly one of the land-grant institutions in the country. And certainly they are the powerhouses of research in the United States, certainly in the area of agriculture. But it gets a little confusing as to exactly what we mean by this division and what we mean by CALS and exactly how the land-grant institution moniker fits in. So first I'm going to, whoops, doesn't look like I have. Oh, there we go. Great. Okay. So one of the things I just want to do very quickly is tell you about the structure of a land-grant institution and the entities that are involved in that. Certainly, we know about the College of Agriculture and Life Sciences, but there are also other entities, and these were created as part of the initial land grant legislations that actually began with the Morrill Act in 1862. Abraham Lincoln signed it in the middle of the Civil War to create institutions of higher learning across the country that focused on science and engineering and educated sort of everyone in the country. And the college is the land grant college and is dedicated to that. But at the same time, additional acts over 20, 30 years created the thing called the Arizona Experiment Station, which was intended to create research for the people. At the time, it was mostly agriculture. Now it's diversified a lot. But for us, that is a tremendous amount of research infrastructure across the state, a variety of locations that range from agricultural field stations to a cyber technology group. So quite diverse. There's also the third pillar, which was created in 1914, which is cooperative extension, which brings science to the people. I won't have time to talk about it, but in Arizona, it's over 1,700 individuals at the university and spread across the state that really provide that link to the people, bringing science to the people. So when we talk about University of Arizona and Bayer, we're really talking about all of these entities coming to bear in our relationship. And I just wanted to make that clear, that it's more than just the College of Agriculture and Life Sciences. It's all of these entities that come together. And we provide infrastructure support also for our new College of Veterinary Medicine, which is really exciting. So one other thing I'd like to mention is our Yuma Center of Excellence in Desert Agriculture. This is an innovative public-private partnership that is in Yuma, Arizona. Of course, Center for winter specialty crop, winter vegetables for much of the country. And this was created to provide a mechanism to be more responsive to the industry needs. I mean, it's challenging sometimes for university and faculty with long-term initiatives and goals to respond on a week or two basis notice to some emergency or some emerging threat. And so this was created. Paul Brierly is the director. He's done a wonderful job, and our partners and the board here are the leaders of those industries, those companies in Yuma and around Yuma, and they apply to a variety of very important topics that come up and sometimes respond very quickly to them. So they create an early and immediate response and allow us to be responsive to the needs of our industry. This is a model we would love to expand and develop in different ways, in new creative ways with other industry partners. So when we look at the university, when the College of Agriculture and Life Sciences, in terms of the research that's performed, this wheel is just sort of showing you the different broad areas we might define the research in across 10 academic units. You'll see that agriculture is actually about 1/3 in terms of research expenditures. And environment is really 41%. So although we're called the College of Agriculture and Life Sciences, we are plurality environmental sciences, which is extremely strong. Some agriculture, quite a bit of biomedical sciences. In fact, if you take a look at research dollars, that's the largest amount. So we are diverse, but we see a lot of benefits in that diversity. And the diversity we consider a strength, because as we think about agriculture and expansion, we think certainly about the environment and how we integrate those. And so as the environment college with much of the environment research on campus, we see these integrations very, very important, even across biomedicine, zoonotic diseases, kinds of pest control, those kinds of things all integrate. So we consider this diversity a particular strength. I'm going to talk today about certain specific areas. I'm going to give 3 or 4 specific areas and go a little bit in depth without knowing in advance exactly what would be most appropriate. And before I did that, I just wanted to list a large number of projects and people that we have in the School of Plant Sciences and Biosystems Engineering who do a tremendous amount of tends to be more of what we call the discovery-based research, which is really the sweet spot of universities historically, is to create knowledge. More and more, we're understanding that we need to create knowledge and move it along the pipeline to application and impact. And we've taken that mandate very, very seriously. But nevertheless, in terms of discovery science, we have a tremendous cohort of faculty who work in a variety of areas that I've listed here. If I've missed anybody who's on the call, I apologize, it's my fault. And you can go to individual department pages to get more information about them. I could talk 10, 15 minutes on each one of these topics. I just don't have time today. As we explore our relationships, we can dive more into these as they become relevant to this relationship. But I am going to talk about several sort of areas that I think will be particularly important, Dan, especially some of the comments you made. The first one I want to talk about is the Maricopa, what we call the Maricopa Field Scanner, which is the largest field phenotyping system in the world. It sits in Maricopa, Arizona, at our agricultural field station there. It is the state-of-the-art phenotyping facility, better crops for a better environment. This is a very large project. It's a bioengineering project. It's an engineering project. It's an agronomics project, and more than anything, it's a data science project. And so when we take a look at this, it's hard to get a feel for the size of this entity, but it is 2 acres under rail, so to speak. And that structure you see there, the gantry, moves back and forth and does very high resolution phenotyping. And it was constructed with a Department of Energy grant initially, with the whole idea of being able to do unprecedented level phenotyping on genetically defined crops to move forward in the idea of gene discovery and phenotype and physiology discovery. Okay, it's quite an expansive project. As I showed on my previous slide, Duke Pauli is the principal investigator for this project and the projects undergoing the gantry. Eric Lyons is integrated in data science, and I'll talk about this just a minute. This was built with a DOE grant, and Pedro Andrade, who's located at our Maricopa Center, was instrumental and really did a phenomenal job in getting this thing constructed. And in some ways, it is a project with multiple different components. And to get an idea of the scale, the box you're looking at there, that's the sensor box, is 10 foot, about 10 feet in length, and it holds a variety of different kinds of sensors. And the way to think about this particular facility, it's like having the largest telescope in the world or the largest mass spectrometer or microscope. It is such a specialized instrument that it provides unique capabilities that are not available anywhere else. And look inside that box, you can see in the upper right, that's kind of a photo looking up into it and showing some of the different sensors and cameras that are available. These include RGB, thermal imaging, fluorescence imaging, 3D, and hyperspectral. And when you put all those together and the capabilities of scanning the field and the diversity panels that we plant under it, its max capacity is to create 10 terabytes of data a day. So you can immediately see this is a big data problem and a data science challenge. And typically, depending on the scan rate, we pull up, it brings off about 1.5 terabytes a day, but nevertheless, getting that data from the facility to a data science environment where it can be processed and analyzed was a significant challenge, and at first was a big challenge to the project. Fortunately, we've been able to solve that one. And that was partly by bringing Eric Lyons into the project. We had an initial data pipeline going to Illinois, but we've since moved it to focus on to move to the University of Arizona and a very large data science project called Cybers, which I'll talk about in just a moment. But what Eric was able to do was to develop with his colleagues to develop a workflow here that could take the data off the gantry, move it to University of Arizona on Internet 2, and then develop a workflow that allows worker nodes and distributed computing to process the data, because each of the data sets needs to get processed. And as you can see, it says what 4 lab computers would take 11 years for, this FIDO Oracle pipeline can do in 1.2 days. So this was quite a data science coup, and we were able to really do that. And that allowed us to move forward in a number of very exciting ways and take advantage of the unique aspects of this facility. And I want to tell you just a little bit about Cybers, because this is a $115 million data so far cyber infrastructure data science project funded by the National Science Foundation to design, deploy, and expand a national cyber infrastructure for life scientists and train scientists in its use. It's funded primarily by the National Science Foundation. It's in its 13th year. It's led by the University of Arizona. For the last 6 years, I've been privileged to be the principal investigator of this project. Eric Lyons will be assuming that role just in the next month or so. We have about 90,000 user accounts around the world. It's freely available. Just to give you an idea of scale, about 7 petabytes of data move in and out of Cybers annually as it's being managed and analyzed. And the thing about Cybers is it's universally available. It provides extremely robust data management, the life cycle of your data, and it connects your data with analysis pipelines and compute. And it solves that sort of zone of that valley of death for scientists and data sciences. I've got my data, now what? And this solves that problem. So I think this could be an interesting area of interaction between Bayer and the university and Cybers. Coming back to the gantry, one of the unique things about the gantry is that it can detect photochemical efficiency at scale. And this is a system where you photo bleach and then take up to 100 images in a second to watch the fluorescence come back. And this gives you a reading of the stress of the opening of the reaction centers, PS2 reaction centers, and the level of stress in a plant. And this provides the ability to take a look, and what you see in blue is the field level response, photo response in a normal situation, and in the yellow you see it in a water stress situation. And so you can see we can document that through this approach and look at stress. And the key here is that this method is extremely sensitive and can detect stress in plants far earlier than you can do so by any other method. So if you think about diversity panels and genes and so on involved in stress response, drought tolerance, this is an incredible capability of this facility. Another area is plant performance predictions. Nathan Handler has been able to develop machine learning algorithms that allow us, without getting into too much detail, this happens to be in lettuce, we're able to predict 35 days out with 95% accuracy what the yield is going to be on that particular crop. So this is another way we're using, by incorporating all of the data that comes off the gantry and machine learning, we're able to do these kinds of predictions. Finally, without getting too much time, we're also working on a completely in silico plant and growth and development environment. This happens to be virtual reality, where you can go through the plants in 3D with virtual reality goggles and pull up data. And I won't get into it too much more, but this is another really exciting way to try to go completely in silico with plant growth and development. Another area I'd just like to talk about briefly is the evolution and management of insect resistance to genetically engineered crops. So this is no surprise to anyone here, and nothing new about BT crops and the ability to use BT to confer resistance. But what Bruce and Eves and Peter Chun have done and their colleagues is to really do a deep dive into the biology and the genomics of BT resistance and really understand at a molecular level what's going on. And we have actually the only pink bollworm colony here that's highly secure here to do those kinds of studies. But the other thing they've done is to take a look over many decades of how you go about using those crops and the kinds of strategies you need. In particular, the notion that you need a refuge of wild type crop in order to maintain that wild type reservoir. So if there are any matings, they don't become homozygous, and therefore you don't end up having resistant animals. To give you an example of how impactful that is, India was not good. They introduced BT cotton, for example. They were resistant for about 10 years, but they did not undergo a refuge strategy. Pink bollworm came back. It's now completely out of control. Arizona, we've been free for 25 years. That's combined with the refuge strategy from 1996 to 2005. 2006 to 18, we underwent a sterile insect release along with the refuge, and it was declared completely eradicated in 2018. Just to give you an idea of the impact of that, it was declared eradicated, eliminated a completely insecticide use, which was very, very high. It saved the growers in this state alone $192 million, and the environmental health and economic impacts of this are tremendous. It's one of the most impactful projects that I know of. And so this is an example of how this very capable group is using a variety of techniques and approaches to understand holistically and molecularly how to approach these issues and make them work for the long term. Another area just want to mention is our plant pathology. I'm just going to highlight Judy Brown here, who is using some really advanced double-stranded therapies for psyllid mortality and abatement of the vector-mediated basically citrus greening, which has cost hundreds, billions of dollars. She's got a technology here to use RNAi, double-stranded RNAi, through feed to put into these animals and then confer basically toxicity and reduction. And this is a really, without getting into too much detail, she's got target candidates she's using, and she's doing a screen, and she's found a variety of targets that will significantly induce psyllid mortality and reduce citrus greening along the way. So this is very, very exciting technology. She's also got the same kind of technology going on in terms of single-stranded RNAs for whitefly and begomavirus gene silencing. And this is an example in crops of taking that, breeding crops with these to be able to express these and having resistance for those two, the pathogen, the virus, and the Bemisia tabaci. So this is another way that we're approaching this. Judy Brown is one of our faculty in the plant pathology group. We have others that have very exciting projects ongoing also. Lastly, I just want to turn to, in a segue, to Marat Katsira, who's going to talk about controlled environment agriculture, circular bioeconomy. This is a big buzzword now towards more sustainable agriculture. It's advanced, it has to do with having this circular economies around agriculture and being able to convert the outputs into inputs in the cycle of the particular product you're talking about. We have ongoing projects in animals in terms of bioenergy and biofuels, in cattle in terms of capturing everything and recycling. I'm not going to talk about that. But certainly in terms of sustainable biomass sourcing and reuse, we've been involved in this one for quite some time and moving this forward. And I apologize for the complexity of this slide, but we have a project called MISES, which is Modular Integrated Sustainable Agriculture Systems. And the basic idea here is you take a variety of organic wastes and you reprocess them so they become the inputs for other activities, in this case for feeding insects, which then provide both organics and also food for the other parts of the cycle, and hydroponics and fish, mushrooms, and leading to new food. The key in the red box is developing ways to process the organic waste. And just to give you an idea of the scale, we've been working with Mexico City on these processes, and Gagi Davidowitz has been heading this up with a team. Every day, their main market throws off 40 tons of organic matter. And the problem is, how do you take that? And most of it ends up in landfills of our food. And the question is, how do you take that and process it before it rots, especially in warm climates? And what Gagi's been working on is development of a large-scale solar dryer that's highly technical. I won't get into it right now, but many sensors, and the net output is water, because some of these food wastes are 99% water. They collect solar, heat sink, water coming out, and you end up with a dried product that then can be an input for insects fed dry food waste to be fed this dry food waste. And insects, and Gagi is an entomologist, he's got exciting things going on in insect development, but the important point is that insects then provide an input both for incredible organic matter in their waste, but also as food for fish and other products that then become inputs into this sort of circular closed environment. And as a segue to Murat, I just want to mention, this is a very busy slide, but it shows you a whole variety of areas in which we are working in the controlled environment. Center is working ranging from mushrooms to space. We have the lunar greenhouse to enter-producing greenhouses, high-value crops and vertical farms, vaccines and natural products, and high-throughput phenotyping inside. So with that, I'm going to let Murat continue talking about controlled environment agriculture. I'll turn it over to Murat. And unfortunately, there we go. Sorry, my mouse. This is awkward.
Thank you.
My mouse
Thank you,
just
Parker.
died. I'm trying to stop sharing and my mouse just, oh, there we go. Good, thank you.
Thank you. Thank you, Parker. Just to check, can you see my full screen?
Yes.
Awesome. Thank you so much, Parker. I appreciate it. And also, thank you for the opportunity, Phil, Alex, and Dan, as well as Stefania, for us to share with you some information about our programs, controlled environment agriculture programs, as part of the AgConnect event. I am super excited, and I look forward to furthering our collaborations with you in the controlled environment agriculture space. I'm with the Biosystems Engineering Department and also the Controlled Environment Agriculture Center. And I have the privilege of working with an excellent, talented, and passionate group of faculty, staff, and students, and volunteers and industry collaborators as we innovate and shape the future of controlled environment agriculture, technologies and systems, and continue educating the future leaders of the controlled environment agriculture industry. I would like to say that modern agriculture should no longer rely only on the availability of natural resources and the challenging local climates, but it should effectively embrace the digital age by smart and innovative technologies, data-driven strategies and practices, really shifting the paradigm from focusing only on producing more to better quality produce with high nutrition, safer and fresher produce for the diverse needs of our people, and with enhanced resource use efficiency, as well as with lower environmental footprint. We are all familiar with the challenges that we face in our region, but this is also common to other regions around the world in terms of water limitations and the pressure that brings to our farmers and ranchers. And they are faced with this challenge to either give up in some cases or to somehow adapt to these challenges with new strategies and technologies. Our food systems have been challenged recently because of the pandemics. And as you can imagine, the effect on the labor can significantly affect the distribution of food and produce to the consumers. Recently, as we were going through the pandemic, our farmers and ranchers were concerned about the impact of the pandemic on their operations, according to this recent survey. And we have all experienced these challenges, the shortage in the food, in some cases, in some locations, but in some localities, the wasting of the produce because the food distribution systems have been disrupted. So what I would like to say that looking towards future to more sustainable agriculture and sustainable future, there will be a need for integrated and complementary smarter production agricultural systems. There will be no single bullet solution, but there will be an adaptive technology as an option and integration of various technologies as we address the challenges for food, for water, and energy. So why controlled environment agriculture? I have a huge list here. I'm not going to go through all of it, but production quality and consistency is one of the significant one. The resource use efficiency in terms of water use, in terms of the nutrient use efficiency is much higher in controlled environments compared to field-based agriculture with certain commodities and produce. And there's again the list here, but I also would like to underline that controlled environment agriculture also captures imagination of young generation. And there has been a huge investment from the ag tech sector for the indoor agriculture systems, including both greenhouses as well as vertical farming based systems. Controlled environment agriculture really addresses and directly there's a direct synergistic towards the majority of the sustainable UN's sustainable development goals, but also the leaps of 10 leaps that are identified by the leaps by Bayer, addressing the cure through microbiome health and environmental impact in agriculture, and also preventing cancer or curing cancer, but also developing sustainable protein supply. I believe controlled environment agriculture technologies and approach can help contributing to address these challenges. The bottom line in any kind of agriculture production system is the resource use efficiency. As Parker addressed, we are interested, yes, in terms of the outcomes, but we are also interested in we are needing to address to the resource use in agriculture production. For controlled environment agriculture, the main objective is to, of course, produce crops with high yield, the edible biomass with optimal resource use, but also non-edible biomass can be converted into valuable inputs, and they can be leveraged as an input and recycled within the system with circular systems, integrated systems approach. Because of the recycling nature of the controlled environment agriculture systems, the nutrients, the water can be also recirculated. The water from the air can be harvested. So these are some of the advantages of controlled environment agriculture systems. Automated crop diagnostics, decision support systems have been advancing and being incorporated into controlled environment agriculture. We have been working on multi-wavelength-based computer vision, machine vision systems for plant health monitoring and using that information directly from the plant in the decision-making to improve the resource use efficiencies in controlled environment agriculture systems. The interest in indoor vertical farming has been gaining, and these are some of the largest vertical farming operations in North America. But recently, if you may also observing these, that traditional field-based agriculture companies are also investing and merging with indoor vertical farming companies because of the need for consistency and quality of the produce that they're needing for their consumers. However, the challenges in indoor vertical farming space has been more on the electrical energy used for lighting as well as cooling and dehumidification. In order to address this challenge, we are funded by the USDA Specialty Research Crop Initiative funding opportunity, collaborating with Michigan State, Ohio State, and Purdue to address these challenges, to co-optimize environmental parameters, design new systems and technologies to address the electrical energy use, the resource use, with improved and engineered systems to help the indoor agriculture, controlled environment agriculture industry. Some of this research work continues here at our vertical farming facility, at the Controlled Environment Agriculture Center campus. Our main emphasis with research is more towards computational modeling and fluid dynamics, as well as experimental studies to analyze the airflow uniformity and then determine alternative designs that can address the non-uniformity of the environment in this three-dimensional space in vertical farming systems, ultimately towards improving resource use efficiency. We are focusing on and developing phytometric, biofeedback-based decision support systems and tools, in a sense, in a way that plants controlling their environments based on their growth and based on the ultimate production objectives. So our research currently looking into, again, modeling, predictive modeling to determine the plant biomass and growth and yield, and use that as a feedback in the system to control the environment and also the lighting for the optimal crop production. We're also looking into strategies, environmental control strategies, as we try to, for example, reduce the light intensity while alleviating the CO2 to achieve the desired yield, leading towards resource savings in indoor vertical farming space. As Parker indicated, we're really interested in integrated circular systems approach, because in some cases, a waste can be an input for the other system. Here, we've been focusing on mushroom production, for example, in indoor settings where the CO2 is a waste product or emitted, which can be harvested and leveraged and used in a plant production system as part of the photosynthesis. And while we're also harvesting the water and humidity from the environment, which can be used on the other side for the mushroom production system. This project was funded through Parker's office through the CAL's IVIP funding opportunity. The phenotype, yes, is a function of the genetics, environment, and management. We have been trying to address the environment and the management variables in this equation through engineered systems, technologies, strategies, and the capabilities of the operators. However, the genetics has to catch up as well. We need new varieties, new breeding varieties that are suited for indoor vertical farming systems, along with the greenhouse-based systems and technologies. And we appreciate the developments and emphasis from various companies around the world, as well as those from Unfold, a company collaborating with Bayer's LEAP and Temasek. That is definitely needed, because we need to bring both biology and as well as engineering to be able to succeed in this area. I would love to see greenhouse systems producing their own energy towards future agrivoltaics, being able to co-produce food and energy within the same footprint. We have been focusing on these photovoltaic technologies with wavelength selective materials, organic photovoltaics, and we have demonstrated that the light use efficiency is much more effective and greater in organic photovoltaic based or photovoltaics based systems in greenhouse operations, without so much sacrificing from the yield, but with the capabilities to enhance the resource use optimization. I see greater potentials in this area as we look at future energy producing greenhouse systems. Of course, there are various questions, research questions. This concept, the idea of off-grid greenhouses, off-grid water treatment systems, are part of our currently funded NSF project through the Indigenous Food, Energy, and Water Sovereignty and Security project, as we are trying to address the water and energy challenges of the indigenous communities, in our case with the Navajo Nation, but also relevant to other indigenous remote communities. We are interested and we are working on other innovative covering materials with quantum dots, for example, to enhance the spectral quality for crop productivity. As part of our circular systems approach, we are trying to leverage waste materials as a substrate that can be used to produce alternative proteins such as mushrooms. And with my colleague, Gagi Davitovich, we're trying to convert the waste material, food waste material, to produce proteins and edible insects for human consumption, as well as as a feed for the aquaculture systems. As we are focusing our research on challenges addressing terrestrial systems, we are also funded through NASA as we try to innovate and develop technologies and techniques that will help us to further and efficiently grow crops in space-based settings. We are funded now through NASA as we are designing and developing an efficient water and nutrient delivery systems for the microgravity environment of the International Space Station. Our team here at SEAC is part of 5 teams competing in this first phase towards the second and third phase in this challenge. We appreciated our collaboration with Bayer recently through my colleague, Dr. Gene Giacomelli, and his team collaborating with our colleagues at Bayer in the seed corn project as they evaluated the capabilities of ebb and flood bench irrigation and fertigation systems, and also the timeliness and reducing the time for production and quality of seed corn. And I believe this the outcome of this project was instrumental and helpful to support the needs of the technology integration for the Bayer Marana Greenhouse facility. We are looking forward to furthering these collaborations. We also offer outreach programs to educate community members, those who are interested in controlled environment agriculture systems. We recently completed our first virtual annual short course on controlled environment agriculture with 230 participants representing 5 continents and 15 countries. And I really appreciate, again, and I would like to acknowledge Bayer's support for that program, and we are looking forward to our continued collaborations. And this was the acknowledgement from the Bayer team. Really appreciate Stephanie Bowes and her team's collaboration with us on that. We are proud of our graduates, and they are putting their skills and their passion to support the operations of the Marana Bayer Greenhouse facility and the team. We are looking forward to furthering that relationship with you, as we are really focused and more determined more than ever to continue educating the future farmers, engineers, and the leaders of the controlled environment agriculture industry. Thank you again for the opportunity. If time allowing, I would be happy to respond to questions.
Well, thank you, Parker and Murat. That was a really, really awesome overview of all the great stuff going on in your little corner of Arizona. I think let's keep going because I think we want to stay on time. But it's now my pleasure to bring the conversation inside of Bayer a little bit and hand over to Stephanie and her team at our Bayer Marana site to give you what I hope will be an exciting tour of the facility there. So, Stephanie, the floor is yours.
Awesome. Thank you so much. And April's getting ready to share our slides. There they are right there. It's so great to see all the innovation at the University of Arizona and to have been a part of some of those projects and to see those now come to life here in Marana. And we're really excited to share that with you today. I'm going to introduce our team really quickly here, and then we'll go through how we do what we do every day. So if we can go to the next slide there, April. All right, so we have a quick overview with April. We'll do automation and greenhouse, and then we will leave some time for questions, hopefully, and feel free to use the chat function. So Megan Dickens is our interim site lead. She's also our operations lead. She's doing double duty for us right now. She is a fantastic storyteller, so she'll take us through our automation space in just a few minutes. April Carroll is our digital strategy lead, and she's going to take us through how we do what we do, and really excited to share that story as well. And then Brad Hart is our agronomy lead. And Brad and I have been lucky enough to be on this project from the very beginning. And so he has some unique stories to share as well about that. But the star of the show for us is the future corn seed that will go into a farmer's field many years down the road. That is the star of the show here at Bayer Marana. And so without further ado, I'm going to toss it over to April to talk a little bit more about what we do. Oh, I think she's trying to unmute.
All right, thank
there you
you.
go.
All right, so we use Teams a bit, so working on Zoom skills as well. So I want to just give a really quick overview of our site and some things maybe that are not so amenable on the ground, but I want to save all the time possible for our tours. So our location is in Marana, just northwest of Tucson, right along the I-10 corridor. And we chose this location as a company. It was kind of a tough decision because it's always easier to add on to an existing facility, but our existing facilities were missing some of the parts and pieces that we really needed to operate a greenhouse at this size and scale. So Tucson was chosen because of the sunny geography and low humidity, the cost-effective utilities, and I have to say the local talent from the U of A has been phenomenal, particularly coming through that SEAC program, but also we're close to Arizona State. So we can bring in talent from there as well as from some of the agriculture production facilities kind of all around in Yuma and otherwise. And last but not least, we wanted a site that was in the continental US that would have kind of more of a stable climate, no hurricanes or tropical storms or things like that. All right, so here is our site from the air. So we're a 7-acre facility under glass. We have broken ground back in 2017, but there was a long, strong partnership with the SEAC and the researchers there going all the way back to 2015. So just to highlight a little bit of kind of the why behind the facility, sustainability is in everything we do, and the facility was constructed in large part to protect the pipeline and get everything indoors, but also to really conserve natural resources. Bayer is all about sustainability, and we're working to kind of optimize our production as well as our seed production, research production and seed production. So this is the equivalent amount of acreage that was grown in the field previously prior to the launch of the Marana facility. So I just want to highlight also kind of our newest, latest, greatest breeding methods. And so over the past 30 years, we've really perfected selective breeding to plant large quantities of seed, evaluate those seed in the field using advanced genomics methods and other methods for selection of the best. So this is largely driven by connecting and digitalization of the pipeline and operations over the last 5 years. So everything having, say, a DNA marker profile, things like that have brought our breeding program and our selections to the next level. But where we want to go with precision breeding in the future, and we're kicking this off, we're fundamentally changing the entire approach to breeding. So instead of selecting the best traits, we're moving to an era where we can actually design what's going to be the best from the very beginning. So starting with the DNA and those markers and those profiles and those phenotypes, really working towards designing the best products based on what we know. So maybe just a little plug for the great work that we've had with the University of Arizona. So we have over 30 Wildcat alums on site. Stephanie is one of those. And we've also got at least 10 that we know of across Bayer Crop Science. So we hope to continue to be an employer of choice for students that graduate from the U of A. Please send them our way. We are always hiring. And last but not least, I want to just maybe give a little more detail to the collaboration that Murat mentioned, looking at sensors, protocols, and environment. The foundational protocols to this to this greenhouse were actually developed in the greenhouses there on the U of A campus. So just a shout out also to Jean, Murat, and Tilak, who really helped to make all of that possible. And so with that, I'm going to toss to Megan Dickens, who is out in the greenhouse and who is going to give us some really, really interesting content out there that she'll talk through.
Awesome. Thank you, April. I want to do a mic check quick because there's a lot of background noise out here. Everybody hear me okay? Okay,
Good to
awesome.
go.
Thank you. So hello, this is really awesome. I have a chance to see some faces virtually that are very familiar to me and I haven't seen in a very long time. So thanks for taking your time today to join here with us in our little corner of Arizona. Murat, I don't know that you could have kind of queued up a better way to intro even just Marana, because the story of this facility is not only about sustainability, it's also that agricultural innovation and taking ag into, you know, the next digital age. But this is also a story about people, and you heard April say a little bit about that. And so I want to take some time, not only here in the automation room, but I know Brad's also got a different flavor once we head out to the greenhouse. I want to take some time to talk about how we've really been very intentional about bringing all three of those components together. And at the very beginning, it starts with a corn seed. And so what I've got in my hand here, though it's much prettier than your average corn seed, what I've got in my hand here are some colored beads sort of representing those unique traits that we're looking for in every one of those individual corn lines that we've already invested a number of years and resources into. This is what arrives on Marana's doorstep and starts to cue the next step in our processes. But I do want to speak to the fact that even just to get these particular and genetically unique seeds into this little O plate are the fingerprints of data scientists, agronomists, even maintenance and facility teams, biologists, research and development, laboratory scientists, and a whole host of other folks that have already invested a lot of time and energy into getting these seeds to where they are today. The really cool part about this O plate also relates to something that April talked to us about, in that we've made very precise selections that reduce the number of seed in general that we need to plant. So every square foot in Marana is being used to grow the best of the best from the very beginning. And just by the nature of the facility, we're able to protect from, you know, earth, wind, and fire, if you will, able to protect the investment in these seeds as they make their way through our facility. And so to start, these seed are in this O plate. What we're going to do first and foremost is get them into some soil. And so I've got one of our germination trays here. And behind me is our seeding line. The first piece of automation that a seed encounters here in Marana is our assured identity seeder, also lovingly referred to as our AIS. It's an in-house piece of automation that not only transfers those very precious seed from the O plate to a germination tray filled with soil, but also does a lot of the heavy lifting from the data. So data is the #2 product that this facility produces, and a really, really important part about our story. Without the data, without that identity, without the genetics, the rest of this really doesn't matter, because we want to be prescriptive in that precision breeding mantra in terms of moving the material through our system. So that assured identity seeder is going to take that seed, drop it in a beautifully filled germination tray full of soil. Our data will be transferred accordingly. The rest of our seeding line does some of the heavy lifting for us. It'll top dress those seed flats, and then it will water from both above and below. And then we will send those germination trays off to our germination rooms. So we've got two kind of mini controlled environments powered by LEDs, where we can also control things like irrigation, nutrient, temperature, lighting, hours of day and intensity, in addition to just general climate settings. And those seedlings will spend about 7 to 10 days there. Once ready, we'll pull them back out and move them on to the next part in our process. And I see April's got a video ready to cue. Don't hit play yet. I do want to talk through a little bit about what you're going to see in the video, and then we'll kind of recap it while you're looking at it. So after 7 to 10 days of that seedling growth, we take those germination trays to the next piece of automation equipment, which is Apex, our automated plant extractor. This is a somewhat off-the-shelf piece of automation that's used fairly widely in floriculture and vegetable production. We've customized it to do the things that we need to do with corn here in our Marana facility. So as we send these germinated seedlings through, inside there's tines that will drop down, extract the seedling from the flat, take an image, and compare that image to some logic that we've provided related to leaf height, area, color, and that piece of equipment will make a decision for us. Does this match the criteria I'm after? Is it close? Or is it not even worth looking at? So if it's not worth looking at, we'll discard that both digitally and physically. If it's close, that piece of automation will send it back into a germination tray where we'll head back to the germination room for just another couple of days until we can bring it back out. Again, right, these seed are selected, they're here for a reason, so we want to try to pull forward everything that we can. Seeds that match or exceed our criteria are automatically transferred into a pot. Every one of our pots has a unique barcode around it, again, keeping track of that unique identity as that seedling moves through our facility. That pot via conveyor is fed into a robot that will pick it up, drop it on a bench, and then ultimately move that bench via overhead cranes and conveyor systems out to the greenhouse. And so April, if you're ready, I'm gonna have you play that video that we can talk to. And through the power of TV magic, we will make a bench appear out in the greenhouse where Brad is waiting for us. So the video right now is just showing a high-level overview of our central automation room that sits right in between both of our greenhouse ranges. The greenhouse ranges sit on the north and south side. We've got two distinct workflows that we're managing here. That physical separation just helps us manage that most effectively. So there you see our germinated seedlings heading into the apex. Those grippers or tines that are dropping down to pick seedlings up out of the tray, drop it in a pot. Conveying that down the line to our pot placement robot. Dropping those on a bench. Once a bench is filled, it heads to one of our two cranes where it is picked up and carried overhead out to the greenhouse, where I believe Brad is patiently waiting to continue talking to us about the rest of our corn life cycle.
Yes, and hopefully everybody can hear me. I'll do a little mic check. It is even noisier out here in the greenhouse.
Sounds
so
great.
Awesome. I will continue yelling, and hopefully the video feed's also working well. So again, we did want to share with you a little view of our greenhouse, and I will echo what Megan said, is we could not have gotten a better introduction from Marat on why we chose to move these workflows into a greenhouse. And even so, why we chose a greenhouse in Arizona. So as I'm talking, you're going to see some stuff moving behind me. And obviously, this is one of the things that the corn breeding pipeline, again, transitioned from these research style greenhouses historically in all parts of the company that we've been using into a production size greenhouse with production systems that are traditionally used in much different industries, but similar at the same time. So the things that you'll actually see moving behind me, so these are our benches. One of the, again, efficient use of our space comes from the fact that where our plants are under glass is almost entirely occupied by the plants themselves. And we actually bring the work to the people. So you can actually see we're on the opposite side from where our workers would be, and we'll actually get to see a video a little bit of what that manual part of the process looks like. But this is, you know, kind of the first step in that being really efficient with the space that we have is use all of the space that's climate controlled and developed for plant growth for the plants. And by using this automated bench movement system, we're able to do that. The other thing that you'll notice is they are moving around on tables here, and these are flood tables. Again, going back to the work that we did at the University of Arizona at the SEAC, really figuring out how to transition all of the knowledge that we had in growing corn in different ways in containerized media to an ebb flood system, so we can recapture about 90% of the water that we use on a daily basis. So really the only water that leaves our system, there's two ways that happens. It's either through cooling, evaporative cooling, again, one of the benefits of being in that dry, arid climate. And then what the plants are using and uptaking and helping cool themselves, obviously. So very efficient with water use, very easy to balance nutrient content and deliver, you know, the right nutrients to the plant. I think one of the main departures from some of the things that we were talking about in the previous discussions is we are the middle part of Bayer's corn breeding process. We are not developing corn lines to be grown in a greenhouse, and we needed to be able to grow all of these lines that are being bred for all over the world as quickly and productively as we possibly can for the short time that they spend in Marana, all while maintaining the data that's associated with them. So again, going back to what Megan said, we know the identity of every seed, we keep the identity of every plant, and we also track the identity of every action that we do to those plants. So again, you really tie in this very robust system to not only need to be a high throughput production system and a high throughput biological system, but also, you know, massive amounts of data analysis and data capture goes into this as well. So I think that was all that we were going to see moving. We might actually get to see the crane here. I just heard the door open up, so maybe we'll give it another second. If not, we did have a video that shows where our people are still very heavily relied on. Again, automation does not replace everything that we do here. We still rely very heavily on people to help us execute the most critical part of that corn breeding process, which is making those pollinations. So we're gonna hit play on the video here and I'll kind of talk through it. But again, you know, making sure that we have the right genetics that get handed off according to the project that the breeder would send to our facility. We do 100% controlled pollinations on every plant that we have. And again, here you can see we cover the flowers, we hand collect the pollen. It's kind of jumping for me, but hopefully it's playing all right for everybody else. But this is again where our data is actually instructing the operators to be able to do this process as prescribed with whatever project there are. And all of the automation tools that we have help support this work to be delivered to the people at the workstation. So we can actually prescribe the benches to arrive in a certain order. If we need to do that, we can actually have them kind of instruct the work that needs to happen. So, and here you can see the actual act of pollination. So the realization of all that genetic testing and data is really in that one step where we have to realize the seed that we would produce. And hopefully the video is ended. I could not see it actually end on my side, but that's all I wanted to share with you. Thank you again for joining us here in the Marana Greenhouse, and I will turn it back over to April.
All right, thank you, Brad. So that is the end of our virtual tour, but we're happy to take questions. Not sure what kind of timing we have left for questions. Maybe Alex or I'll try to read up on questions in the chat if you want to put those in the general chat, or just unmute yourself and ask away.
Yep, we have some. Sorry, April, I was on mute. We have some time for questions for either you guys or the U of A team.
So I have a quick question. This is Jay Petrick from Bayer. How much of the energy use at the Marana Greenhouse is solar versus the local grid?
So the energy use right now, we are all on the local grid, but we do have some really ambitious carbon reduction goals. By the year 2030, we need to be carbon neutral. So we are looking hard and fast at where we might be able to reduce our carbon footprint. And Stephanie has done a great job working with with the scientists within Bayer, as well as doing some research on how we can reduce that. But if you have other ideas, happy to hear them.
Very impressive. I'm in Tucson often, so I hope to come out and check out the greenhouse again.
Maybe to add on that, April, too, is that as it gets back into the biology side of this, this is also some of the longer term plans. So not only to choose where our power is coming from, but also the technologies that we're using in our greenhouse to help reduce the power. So like Megan said, in our germination space, we take full advantage of LEDs. One of our hopes and long-term plans is also to get the right LED technology to use out in the greenhouse as well. So again, that sustainability goal and carbon neutrality goal kind of percolates through all of our short-term and long-term plans for the greenhouse here.
This is Eric Lyons. I got a question. Very curious on the data side. If you can talk at all about how much data you're generating. Are you processing it on site? Are you shipping it all back to St. Louis or beyond? And just a little bit curious about how your teams operate, because I know you're distributed around the world.
Yeah. That's a great question, Eric. And I think last time I saw you, we met at Phenome, if you recall. So my focus area around data is all about how to get actionable decisions out of the millions of data points that we collect. And I think you know better than anyone how difficult it is to kind of wade through all of the information to really try to get something actionable out of it. So I'm not even sure how much data we generate, but I guess to your point, it was enough that we had initially started off with a cloud-based system, and we realized pretty quickly that we couldn't get the speed we needed by constantly having to upload and download information from the cloud, particularly about our operations. So we did make a big decision a little over a year ago to kind of host our data on site. And so we're doing a great deal of the processing happens on site. And we do good, probably 95% of the analytics as well, all go on site, but we collaborate heavily. Say, for example, data scientists back in St. Louis help us out with some operational efficiency modeling. And we have really deep support with the teams in St. Louis and all around the world that are doing similar type work that have a lot of experience on that. So we're very well supported. And even during COVID, we were very well supported online. So there's a question here in the chat. Is the climate the same in all the greenhouses, or do you change it to evaluate different lines? Brad, you want to take this one?
Yeah, so good question. It looks like that's Neil. Hi, Neil. But we do use different climates. It's again, not to necessarily learn information about the different lines that we have. It's more about to get the best performance out of the developmental stages throughout the process. So again, we actually deal with tens of thousands of lines every year. So we know that we won't be able to come up with a, or be able to tailor an individual climate related to each one of those lines. What we have to find is kind of that peak performance for most of the lines. And how we do that is to really treat the different developmental stages with different environmental conditions. So we do have 13 environmentally controllable compartments on site, and we do use those to our advantage throughout the process to give those different ages of corn different environmental treatments.
Hey Brad, this is Gene.
Hey Gene.
You're looking good there, buddy. And that's a beautiful facility back there. It's great to see the commercialization. My question is going to be about how has the success improved your ability to do what you want to do? Your goal is not automation. Your goal is breeding and doing it more quickly and saving time and energy, basically. Is there anything about this system or any specifics that you would be able to say that you would like to improve upon the design?
Yeah, so maybe I'll tee it up and then I might pass it over to Megan. I think as you think about, again, where we are in Bayer's breeding process, our main goals are to be able to hand off all of the projects that come to us. So again, that percent success that maybe a traditional field workflow doesn't get. That's obviously one of the main benefits of having a greenhouse. I think the other thing that we're starting to realize and have the potential to realize is the speed at which we're able to do that and the performance that we're able to get out of individuals. So again, if we're able to input less, output more, then obviously that's where we really see protected culture kind of culminating those three areas and making our system a lot more efficient. And that kind of goes back to one of those slides that April had, where this workflow was about 200 acres in a field. And we're at 7 acres here on site. So just from the space resourcing, we get that. And if we start to realize some of the time and production efficiencies that protected culture has the ability to deliver, then that would be really impactful too. And I don't know, Megan, if you had anything else to add to that.
Yeah, I'll jump in and maybe try to answer another one that was about how many cycles have you completed. So we opened our doors in March of 2020. And the beauty of a protected culture facility is that you can plant every day of the year. So though this might seem like a crazy number, you know, we're somewhere around like 60 to 75 crop cycles if you look at it on that week by week basis. Now, some of those were very small as we're in scale up to production operations, but still so many shots on goal and great opportunities every week to learn something new about the system. And Jean, to your question, you know, I put my production operations lead hat on here. And unlike a field where you can deploy people down rows and in different parcels of acreage, we have to think very differently about how we execute. So what you see behind Brad, there's a very limited amount of space on the other end of that greenhouse where the people do all the things. Brad talked about pollination, but there's also maintenance, there's bench movement, there's integrated pest management, there's irrigation scouting. It takes about an hour for a bench to get from one end of the greenhouse to the other. So every time we have that bench of material up on a workstation where the people can interact with it is incredibly valuable. And so how we orchestrate the flow of movement for all the things that we do here, it has been, I think in my mind, a huge learning opportunity, but to see how the people that we have here with the unique experiences and skills come together to solve that problem really quickly and very effectively has been like insanely amazing to be a part of.
There's another question in here. Kelly asked, do you use proprietary software or off-the-shelf data collection applications? So we use a combination. We try to buy things off the shelf where we can and where it makes sense. There's no need to reinvent the wheel if there's already something out there. But we did, for data collection, and because we have to integrate with some legacy systems within our breeding organization, we did build our own data collection application. And so one of the biggest challenges that I go through most days is figuring out how to integrate the data streams from the third-party systems into our proprietary systems that really eventually house all the information about the breeding pipeline. So great question. There's another question here. Are there challenges with water recycling, disease, or plant nutrition? Megan or Brad, if you want to take that one.
Yeah, I think this is where we, you know, again, had the fortunate opportunity to kind of learn all the hard stuff at the University of Arizona. So, you know, we got, I don't know how many years it was of testing, again, moving to a very different watering program than we use in other places. The only, you know, real challenges that we've had are, again, with some of the scale of the operations that we deal with compared to what we tested on. So, our water holding capacity here on site's about 500,000 gallons that we use every day. And again, about 90% of that's recaptured. Very talented people coming out of the University of Arizona on our growing team that, you know, have kind of eliminated a lot of the major concerns that we've had with water chemistry, fertilizer delivery to the crop, and even some of the systems that we have to sanitize the water. So again, the real benefit there of kind of learning all the hard stuff with the experts helping us kind of made the startup a lot easier for us, and we've been reaping the benefits ever since.
All right, and we have time for just one last question. So I'm going to read this one about, is there interest in exploring indoor scouting? So with means other than drones, which we did sponsor a senior design project for the UA engineering. Brad, are you talking?
No, I was just shaking my head. Yep.
Okay.
Yeah, I don't know. April, obviously you could jump in here too. I think
okay.
As we're operating the greenhouse system, the more passive data that we can collect, again, because we're not growing a single crop that's designed to be grown in a greenhouse. It's really the nature of our system, and all of our data collection is to try and glean as much data for the small amount of time that we actually get to interact with the crop. We are pursuing a lot of technologies that can tell us more about our crop health. So we're very interested in those technologies and have the teams of people both on site and even within the company that are focused on that. So very interested in it here.
All right. Well, with that, Stephanie, you want to close out?
Yeah, just another quick thank you to all of our partners at the University of Arizona. I did have to correct April that I'm actually a Sun Devil from Arizona State, but I am a huge supporter of the University of Arizona all the way up until football season, and then I have to I have to throw it behind my Sun Devils.
Do that? I apologize to both sides.
No worries. No, right? I know. So Phil was just telling me that his Zoom has died on him. And so I think what I will try to do is pass the ball to our next speaker while we wait for Phil to get back in. And so I am trying to pull up information, and I'm just gonna biff this, and I'm really sorry. But I know this is a very cool speaker that we've been waiting for Space for Food from Dan. Are you on the line, Dan?
I am. Can you hear me?
I can. Take
Great.
it away, sir.
All right, and I'm going to share my screen. I believe you should be able to see a presentation from me, right? Hi, everybody, and thank you so much for making a little bit of time. It certainly has been a lot of content that's been really exciting. My slides are doing that thing where they're advancing before I'm ready for them. I help Space for Food launch, and Space for Food is an initiative by the Yield Lab Institute. Yield Lab Institute is based in St. Louis, but they have operations, and they do open innovation challenges in food and ag, especially around sustainability all around the world. They have operations on multiple continents. And we saw some of the things, this is a very timely thing. So some of the things that actually we heard talked about from the CEAC and from the Bayer presentation are things that we've seen. And there's this real understanding that solving the problems of food in space actually coincides with solving the problems of food on Earth. And so the idea was to, while there's a focus on how humans are moving into space and how they're going to be fed there, let's make sure that those innovations translate both back to Earth and that the earthbound innovations translate to space. So the reason that this matters, the CEAC presentation is perfect for this, talked a lot about this, but what we see is, space matters because as people move into space, ag innovation on Earth and ag innovation in space and food innovation in both places really are very similar, have similar needs. In fact, if you think about it, as humans move into space, the ISS or a lunar base or whatever is just a much more extreme version of a food desert that we would have on Earth. There's, food security is, and the need for access and equity and all of those things are even more important because, as we joke, if the lunar base crop fails, you're not going to call Instacart to suddenly restock the moon base. So they're going to, we're going to have to figure out how to do that. And some of those areas are, ag innovation in both places, but, on Earth, we know it needs less waste, less resource extraction, needs to be closer to the consumer in many cases. And things like, we heard from the CEAC presentation, why controlled environment ag is becoming more important. And, consumers are caring about things like cultured meat, and all of those things are emerging. And then when we look at food innovation is really around security and equity and access. And there's a need for supply chain innovation and product innovation that's really about things like personalized nutrition and, some of the more values-based things like plant-based meats and a lot of the changes that are going on. Well, when you look at what space has to solve, they have to solve for circular systems. They can't afford that. There is no aquifer to tap, in orbit. So they have to figure out how to be really efficient with water. They have to figure out what they call ISRU, which is in situ research utilization. And how do we turn this sterile soil on the moon or the Mars into something that can be organic and help produce food? So the constraints are really aligned, but one difference is that it's much higher profile right now. And there's an impetus to solving these problems, and there's some increasing capital flows into it. So what we really started Space for Food with is, let's look at this opportunity to explore space. And when you see what CEAC has been doing, especially when you look at, Biosphere 2 and the lunar laboratory that they already run, you can see how looking at this area of innovation, extends across both. So we started Space for Food to figure out how to serve these emerging ecosystems, make sure that these people that are creating innovations get supported. Specifically what matters to you guys today is we're launching in this fall the Space for Food Student Hackathon. And actually, we've had some great, Murat, we've had some great conversations with Jean and some of your students that have really helped us think through how to do a hackathon that allows people to explore these ideas of these really circular, really low waste food systems. And the example of what these hackathon challenges might be are: propose a food system for a group of 20 or 30, or more lunar or Mars explorers that could be built in 2031 that is as circular, equitable, secure, and nutritious as possible. And propose the same thing for a community on Earth of 10,000 people living within a 5-mile radius. And the goal here is not to say that we're going to, in a hackathon, solve a problem that nobody else has been able to solve. I mean, we've certainly seen how much talent is working on this. But what we do need is people, the next generation of people, to enter these fields and to understand why CEA and all of these fields really matter, and to have their first experience. So we're putting together this hackathon where they're going to be able to meet with people and build their own network, and to actually learn and kind of do their first project in this space that whets their appetite for doing this going forward. So we're looking for high school and undergraduate students as participants and team leaders, but we're going to be offering some mentorship so that the team leaders get to be able to say, I brought together a team, I helped organize them and rally them. But at the same time, we want to have the grad students and the postdocs and the early career professionals have an opportunity to participate. And so we're going to let them be mentors to guide a team and to help provide subject matter expertise and to give them feedback, to let them also stretch themselves in what is it like to lead another group of people, to facilitate other people developing their potential. And then for the subject matter experts, most of the people that are on here are qualify as SMEs. The early to late career academics and people in the professional career who can give them feedback. So if somebody proposes a vertical farm that's 10 miles high because they think that's really cool, that they can push back and say, here's why that actually doesn't make sense, or ask questions. Why did you choose those crops? Why didn't you do this? And to be able to help them improve what they're doing. So the activities around it are, we're going to have some team leader training. We're going to have a boot camp with those SMEs, and we're actually going to give people an opportunity to move around these, develop in these fields. So we've been talking to a lot of people about how do we give people, these students, an early start in this subject matter, and get it so that they can learn something that they can share with their team and they can use it in experiential learning. And then we'll announce the actual challenges. So the 2 challenges I showed you earlier are just sample challenges. But we'll actually announce specific ones that we develop with our group of mentors and subject matter experts and, you know, all of the other people that are helping us develop this. And then they'll get persistent virtual workrooms that they get to work in all over the world. And we'll supply subject matter experts that can drop in and hear out what they're saying. And, you know, like the folks from Bayer, I mean, it's a great example of the kinds of expertise that exist in a place for somebody to say, well, we're going to have a robotic farm. Really? What do you think that means? Because here's what a robotic farm in 2021 looks like. What does it look like to have one now? Or whatever that is, but to give them feedback and to help them improve their pitch. And then we'll have a demo day to really showcase these people. But the goal is to get these people to, by the time this is done, for them to have met multiple people from multiple academic programs, from companies that they might work with, and to build professional relationships so they pursue these kinds of initiatives long term. And at the end of it, when we do a demo day, we'll have a showcase where we can bring all those people together and let everybody see what this talent base looks like going forward. So that's really it. If you go to our website, it's just spaceforfood.org, and on there there's a link to a page called the Hackathon page, and people can sign up. We're now collecting names of people that want to be involved, but we're also reaching out to people that really care about this, that they care about talent development, and they care about talent development, you know, for this new emerging world that we think is really exciting. And so we hope that all of you find a way to participate, or if not, you know, to share this with other people. And if you have any questions, I didn't put it on the slide here, but I'm just, you can just reach me at contact@spaceforfood.org, or you can reach me through the website, but we'd love to have everybody on this call involved, and it's open to students anywhere in the world. So Gene, I hope that I've done justice to all the conversations we've had with you and your students and everything, but you guys have been certainly very helpful in understanding how did we develop not just the workforce, but develop the interest in pursuing this and really innovating in the ways that we need to innovate. So thanks so much, Stephanie, for stepping up and introducing me. And Phil, I hope your Zoom is back.
Well, it is so funny, Dan. It was up and now it's gone again. So you get me one more time. But thank you so much for this great information. I put it in the chat that I really will share this with all the high school teachers that I work with in the area. I think they'll be really excited to see this. And what a great opportunity, and to really tackle those big challenges that literally everyone on this phone call was talking about today. And so inspiring that next generation, that's super exciting.
Well, I'm thinking about how do we get them in those programs so that they can pursue this. You know, we have to whet their appetite, and many of them aren't even aware that this exists. So this is a great way to just get them exposed. And as some of the folks in the, you know, on the cultured meat side say, we need to get them their first experience. We need to get them their first experience with a CEA so they know what the potential is.
Very, very cool. And then they can come work here someday. All right. Well, Phil offers his apologies that his Zoom didn't work for him well today. I want everyone on the call to head over to LinkedIn to the Life Hub page and make sure you're following that so that you can join us for more of these AgConnects. Bayer really went virtual for COVID, you know, taking those in-person networking opportunities that were literally just happening in Boston and expanding this out across the globe. And it has been really cool to see this happen. And I know this is going to continue. And if you have a topic you'd like to suggest, I'm sure that Phil would love to hear from you as well. And thanks everybody for joining today. It was such a great sharing. And if you have any further questions, I actually will go ahead and put my email in the chat. And I think Phil's was in there maybe earlier. And you can always go to our website as well. And there's a form there that you can fill out if you need to get in touch with me. So thanks everybody. I hope you have a great rest of your day. And thank you so much to Alex and Karen at the University of Arizona for setting this all up for us today. Have a great day, everyone.
Thank you very much.
Bye.
Thank you.
Thank you all very much.
Thank you. Very good.